So today we're going to be looking at population analysis techniques that you could be using in the field one day, particularly if you decide to do biology in years 11 or 12. So for today's discussion, we're going to be looking primarily at quadrats and transects. These are designed to look at distribution and abundance of organisms in particular regions of an ecosystem. So let's say for year 11, you have been tasked with going to a local ecosystem to perform a field study. Okay, your teacher's taking down a pH indicator, they're taking down water conductivity, Secchi discs, soil analysis triangles, the works. However, they've also packed in a ruler and a trundle wheel and told you you need to bring graph paper and a pencil. Odds are they want you to do these two things. All right, so let's look at what they're used for. A quadrat is designed to sample a small area with a lot of organisms that are primarily stationary. Now, that's not to say they're rooted to the spot, but it means they don't travel a great deal of distance. For instance, something like an ant's nest or a beetle or whatnot. And while they can travel, that is not to say they're going to be traveling from here to Sydney, all right, or Melbourne. Now, if you're doing something like this in the field, and you've only got the ruler rather than, your actual formal 1 meter square, it's easy to do. All you have to do, get the 1 meter ruler, like I'm doing here, and put something like a rock, or let's see if we can move it into frame. Here we are. So like 2 rocks to indicate each corner. And take the ruler, swing it around, put a rock in that corner, and do the same for that corner, okay? You've got your corners lined out. Anything that falls within those 4 objects, you draw on your graph paper, and you preferably do it to scale. So if you have it, let's say you've got a 10 centimeter by 10 centimeter box on your page, all right? And you draw everything as you see it inside the box. Now underneath it, you might have a key. Because like me, you're a terrible artist, okay? All the stuff I've done, I've actually paid people to do that for me, or I did it roughly on the back of a napkin and it looked great that one time. So, in your key, you'd identify the particular things. Preferably, you look at living organisms more than anything. If you find a beer can there, you draw the beer can, but it's not that important compared to, say, a particular species of beetle or a particular weed, all right? Now, once you have that, you can calculate the square area that that object takes up. And if you look at our video on surface area versus volume, we know that we can scale this up to represent the ecosystem. Now, that's not to say that we are going to have an exact idea of what's covering the entire area, but rather it's a pretty good average. So, for instance, if you want to do a quick activity, can you guess how many holes there are in the ceiling? It's obviously impossible to count them all because you'd be insane to do so. So instead, you do what I did when I was a kid. I look at the ceiling, I pick an area. I count how many dots there are across, how many are there long, then I multiply them. Then I multiply that by how many areas there are. So I think in my old maths classroom when I was in year 10, there was approximately 450,000 dots, just as a rough estimate. Okay, that's not to say I'd be right, but I'd be pretty close to it. Same thing happens with our quadrats. Now, as a rule, you need to perform a quadrat at least 4 times. 3 is the minimum. 4, a little bit better. All right, and it's best to perform this in a variety of areas so you've got a pretty consistent idea of what's in that region. Now, transects are a little bit different. Transects are mainly used for bigger plants, like trees and bushes and shrubs and stuff. So, if I start at a particular location, I'm going to use my trundle wheel and measure outwards. Now, anytime I come across a particular plant, I'm going to document it. And I'm going to document a particular point of information about that plant. For instance, I might use an initial or a symbol at the top of here to represent, what species it is. So over here, circles indicate river red gum trees. Now, if you don't know what type of plant you've got, just put the symbol down, then take a photo of it. Then later on, you can research what that photo is. Now, also, I put numbers here to indicate how tall that plant might be. So for instance, 7 meters along, I came across a 20-meter tall river red gum tree. At 9 meters, I came across an 11-meter tall river red gum tree, and so on and so forth. The distance you have to travel, your teacher's probably going to specify. If they don't, do probably somewhere between 20 and 50 meters. But ensure you repeat it, again, at least 4 times in different areas. Okay? Now, if you're doing this in a particular ecosystem, you might take this opportunity to test the theory. For instance, in Australia, we know that many plants need water in order to survive. All plants need water to survive. However, in Australia, it's a very dry country. So one would assume that there are more plants where there is more water. We can prove that with this. With your transect, if you're near any form of water source, for instance, it could be a lake, it could be a river, it could be a billabong, whatever. Take this opportunity to start at the edge of the river or the water source and move outwards. What you predict you'd see is that the number of plants would start to drop off. Their distribution would not be equal. So if the distribution is higher near the water than away from the water, we know that our theory is correct. More plants are going to grow near water. Conversely, if we have an even distribution of plants, so there's no high density, low density areas wherever, then that goes against our theory, that plants do not need to be near a water source in order to grow. So their abundance is evenly distributed. All right? Now, once you get back to school, odds are your teacher's going to ask you to compile this information from raw data into an actual graph that goes well with an assessment. Now, with your quadrats, you'd be asked to calculate the square surface area of a particular plant or a particular species of organism, and that's easy enough. You just do some basic math stuff. With transects, we can go a little bit further and turn this into tables. For instance, this one over here is a scatter plot of our 3 transects. 1, 2, 3, and well, let's add a 4th one, because why not? So our scatter plots are overlaid using different symbols. And underneath that, you'd have a key. So, all of the triangles represent transect 1, all of the x's, transect 2, and so on and so forth. Down at the bottom here, we have the distance that we measured on the ground. Over here, we have the height of the individual plants. So as you can see from the plot, there are more plants situated in this area than over here, which indicates to me that, there are more plants growing near a water source, obviously. Okay, that helps. So, and looking at all this information, we're trying to use it to provide evidence of measuring biotic factors. We want to understand exactly what is happening to our organisms within the ecosystem, and are there trends? So, not only are we measuring distribution, which is where they're located, and abundance, how many there are, but these can also be used, if they're done repeatedly over a long period of time, to indicate emigration and immigration, any organisms that are leaving the area or coming into it. You can look at birth and death rates. You can look at the number of organisms that have atypical genetic traits, such as different colored eyes or unique patterning. Okay? Now, if you go out into the field, odds are that you're only going to be there for the one day. And that means you only have a limited amount of time to gather this information. The best advice I can give you is, if you're working as part of a team, to collect the information together by dividing it. So you'd have one person or 2 people performing quadrats. One person who would be measuring out the area, the other person acting as scribe. Same with performing a transect. Okay, one person scribes, the other person measures and observes. Now, if you do this, and then you can collaborate all of your information together, your teacher's not going to be too fussed about it because essentially you're gathering the same information. It's much like how you read with another person from the same textbook. No one's going to be annoyed about that. Now, there is a limitation to this. Depending upon the time of year you do it, you're going to find abundances are going to be different. So let's say your school goes out to this particular ecosystem during the early parts of the year. So January, February, March. Okay, that time we still have a lot of the summer heat. So there's going to be certain organisms that are going to be more prevalent than others. Now, if you decide going, June, July, August, that might be completely reversed. For instance, if you go to a wetland area where you have a lot of migratory birds, these birds would impact on how many insects or local forms of seed life is available. More birds means less prey. Okay? So, if your teacher asks you to do an analysis of this, that's one thing you could definitely bring up. Aside from the fact of, I didn't really understand what we're doing, or I had not much practice doing this, you can do this at home. It is super easy to do it, okay? You just need to practice it. It's like tying your shoes. It was really difficult when you're in kindergarten. Now you could probably do it at 3 in the morning when you're half asleep. So, that's everything about distribution and abundance testing using quadrats and transects. If you have any questions about anything we covered in this video, please ensure to hit us up on Lab of Legends on Instagram and leave a message, okay? Ask questions if you need to. Make requests about future videos and what things you want to cover. As always, I've been Forry, you've been amazing, I'll see you around.